Hi, let's go ahead and start with nerve impulse generation and look at action potentials in neurons and the information travel between neurons. Here we see a neuron, and you'll notice there is a cell body with dendrites and an axon leading away, two axon terminals, and the synaptic end bulbs. The synaptic end bulbs produce neurotransmitter that will send the signal to another neuron. The cell bodies and dendrites are what we call the receptive segment. This is the segment where a neurotransmitter will bind to chemically gated ion channels. So here in the dendrites and also on the cell body will be these chemically gated ion channels. And when the neurotransmitter binds, we end up with a graded potential, just like we had the end plate potential with a muscle fiber. Next is the initial segment, and this occurs at the axon hillock. That was part of the axon you learned, the very beginning part. And this is where the graded potentials that are occurring in the dendrites and the cell body will be summated or added together. And if they reach threshold, we end up with an action potential initiating here. Then we get propagation of the action potential in the conductive segment. The conductive segment has voltage-gated sodium and potassium channels. So when the sodium channels open, we end up with positive charges moving in. And when the potassium gates open, those potassium charges move out. So we have both of those. And you'll notice here where it says receptive segment, we can see what type of channels are here. Initial segment where it's summated, the type of channels we have, which is the same as the conductive segment. In the transmissive segment, this would be in the end bulbs, we have voltage-gated calcium channels. If you remember back in the neuromuscular junction, we saw calcium moves in when the action potential reaches the end bulb. And the voltage-gated calcium channels, when they open, calcium moves into the end bulb and it initiates the release of neurotransmitter from the synaptic vesicles. When you're looking at this, you want to know the different segments of the neuron. The receptive segment, the dendrite and cell body, the initial segment, the axon hillock, and also the conductive segment where the action potential is propagated due to the opening of sodium and potassium gates, and then the transmissive segment where the action potential will cause release of a neurotransmitter. And this neurotransmitter may be acetylcholine, or it could be other neurotransmitters, and we will look at others. One thing you want to know then is the distribution of these different ion channels in the plasma membrane of the neuron. Make sure you know where the voltage-gated channels are. Make sure you know where the chemically gated channels are. Voltage is potential energy. It's the amount of difference in electrical charge between two places. That's why when we have ions on both sides of the membrane, we have a electrical charge or voltage. And this will be measured in neurons in millivolts. Make sure you know how to show the distribution of ions on both sides of the membrane. We've done this before in class where we drew a circle representing the cell membrane, and then we show where there's more sodium and more potassium, where there's more chloride ions and more protein and phosphate anions. There is an electrical charge difference across the membrane of a neuron, and that's slightly different than what we see with a muscle sarcolemma. So the resting membrane potential we're starting with is negative 70 millivolts. At the receptive segment, when the neurotransmitter binds, it will trigger what we call a postsynaptic potential. An end plate potential is a type of postsynaptic potential. The neurotransmitter will bind to chemically gated ion channels and open them. Ions then diffuse across the membrane, changing its electrical potential, and the voltage change then we call a graded potential. It can vary in size depending on the number of ions that diffuse across the membrane. It's a local potential, which means it starts at the dendrites or cell body, but it doesn't go far. It will trigger the initial segment, but it doesn't go all the way to the end of an axon. So the receptive segment is only the dendrite and the cell body. Here it's showing at rest, the membrane potential is negative 70 millivolts. And we can see chemically gated ion channels and also then the voltage gated ion channels in the initial segment and in the axon. But I want to point out to you there's more than one type of chemically gated ion channel in a neuron. We can have a chemically gated potassium channel. We can have a chemically gated chloride channel, and we could have a chemically gated sodium ion channel. So we're going to look at those. But just like before, we're going to see the resting membrane potential needs to go up to reach threshold before we can get an action potential. So we see in this first portion here an unstimulated cell. This is showing what happens when sodium moves into the cell, where it reaches threshold around a negative 55, and then we can see depolarization and repolarization back to our original resting membrane potential. So as we're looking at this, just keep in mind, just like with the muscle, we have a threshold value. The direction of the potential depends on what type of ion channel opens. If you look back at your picture you drew of the cell membrane and where sodium is in greater amount, it should be outside the cell. So if sodium channels open and sodium diffuses in, we're going to have more positive charges going in. We say that it's less negative, or in other words, it's going back up toward threshold. So when we say it's less negative, what we mean is when sodium opens, it's not as negative and it's going up toward that negative 55 threshold value. If chloride channels open and chloride diffuses in, more negative charges are diffusing in, and the membrane becomes more negative. So in this case, if chloride channels open, realize that this is going to go down away from threshold, and that means it's a inhibitory type of local potential. It's going down this way. If potassium channels open, remember potassium's in greater amount inside the cell, that would cause more potassium to diffuse out, and therefore it also becomes more negative inside the cell as positive charges leave. If potassium moves into the cell, we still have more positive charges going out, so the resting membrane potential is going to become more negative. You have to think about that, but it does mention that over here. And so when the cell is less negative, the resting membrane potential is depolarized. So the only one here that would depolarize it, or in other words, cause it to become less negative, would be sodium moving in. And remember, this is a short-lived potential, and so it only lasts for a few milliseconds, and it could lead then, if there's enough sodium going in, to threshold and therefore a depolarization as those voltage-gated channels open. We call the local potential where the potential moves less negative or towards threshold an EPSP, which stands for an excitable postsynaptic potential. And if the sodium moves into the cell, then we see it goes more toward the threshold value. If potassium leaves the cell, then we have more positive charges leaving, and so the inside becomes more negative. Or if we have negative charges moving in, the inside becomes more negative. And that's this example here where it's talking about an inhibitory postsynaptic potential. And what an inhibitory postsynaptic potential means is it's going away from threshold, and it's less likely then to cause an action potential. All of this is taking place here at the synapse. This is showing a presynaptic neuron, and here is the postsynaptic neuron. So excitatory postsynaptic potentials are depolarizations locally that are caused by sodium moving through a chemically gated ion channel when a neurotransmitter binds to the protein, just like we saw this at the motor end plate. So it goes away from, now this one didn't get to threshold, but if we have enough of these excitatory postsynaptic potentials, it could get to threshold and cause an action potential in the next segment. An inhibitory postsynaptic potential, on the other hand, is going to cause this to become more negative and therefore go down toward negative 80, and it's not going to have an action potential. The problem comes in when we actually look at a neuron, that there's many of these synaptic end bulbs coming into contact with one postsynaptic neuron, and each one of these can be either excitatory or inhibitory. So we need to keep track of these. That's why we call it summation, because if we have more inhibitory than excitatory postsynaptic potentials, then maybe the whole potential will be inhibited, and we will not have an action potential here at the postsynaptic neuron. If we have enough, we could have a action potential if it gets to threshold. So if we have enough positive excitatory postsynaptic potentials, then that could lead to an action potential if all of those are summed up here at the initial segment, and then they open these voltage-gated ion channels sequentially, like remember we talked about the domino effect where one drops and then opens up the next one all along this neuron. So we really have then summation of these postsynaptic potentials, and if it gets to threshold at the axon hillock, then the voltage-gated channels will open and we'll get depolarization, and an action potential will be generated. We have what we call an all or none law. That says that if threshold is reached and the action potential is generated and propagated down the axon, it will do this without any loss of intensity. So it goes all the way down the axon, like if you started one domino, the rest of them fall down all the way to the end with the same intensity of response as we had at the beginning. So you want to compare the two of those, and you want to know that we do have summation taking place here at that initial segment. The action potential then involves the conductive segment, and this is where we have depolarization and repolarization occurring. Depolarization is a gain of positive charge as sodium enters through, and repolarization is a return to negative potential as potassium exit through these voltage-gated channels, and that occurs in the axon only. And then the action potential is propagated down the axon all the way to the synaptic knob, or what we call the end bulbs. These involve voltage-gated channels that open sequentially all the way down to the cell membrane of the axon, and then it's propagated, and it's called a nerve signal. As one gate opens, it's going to cause sodium to move in, and as sodium moves in, it becomes more positive in charge, which opens the next gate, and more sodium moves in, which opens the next gate, and so on and so on. So we end up getting what we call a threshold value, a depolarization, and a repolarization, and that's what this is referring to here. That's all I'm going to record right now. I hope that helped. Make sure you look back at the neuron. That's going to answer a lot of the questions you have on the practice questions at the end and the assignment that's due.